Hello students, so here today we are discussing about IGCSE biology and our topic is digestion. We are actually going to discuss the topicals related to digestion. So here I have a question. What does it ask? It asks as figure shows part of the thoracic and abdominal cavities of a human. So here thoracic cavity and abdominal cavity is given, and they have asked us a few questions. So what are they asking? They are asking name the structures labeled G, H and M. So G is this, which is actually esophagus, and H is what? H is this diaphragm, which is actually separating thoracic cavity and abdominal cavity. Okay, diaphragm is not the part of digestive system. And then what do we have? We have M, which is actually large intestine or colon. So make sure that you are writing the correct spellings of these labelings. Okay, then after that let's move on to the next part of the question. So what are they asking in the next part? In the next part they are asking table shows 5 functions of organs in the abdominal cavity. What are they asking? Complete the table by naming the organ that carries out each function. Here we have to name it and then we have to write the letter. So naming the organ that carries out each function. So where the function is carried out, okay, not where it is being produced or released. Like for example, where we are talking about conversion of glucose to glycogen, we know glycogen is a hormone. Glycogen and insulin, okay, they are produced by pancreas, but since they are asking for carrying out the function, so where does this conversion happen? We cannot name pancreas here. Okay, so conversion, it happens actually where it happens here in it happens in liver. Okay, so I am going to write liver over here. And what is the letter? Letter you have to see in the diagram. So should I take you back over there? Okay, so you have to find the letter for liver. Okay, so for liver, where is the letter? Letter is actually P. So what will be my answer? My answer will be P here. Okay, so this will be my answer, liver, and the letter is going to be P. Understand? Okay, after that what do we have? Okay, let me move it a bit here. Then after that, the second one is already done for us. Secretion of insulin and glucagon, as I just said, that it is produced in pancreas. It is secreted by pancreas. So this is already done. Okay. After that, absorption of products of digestion. Whenever we are we think about absorption, what comes to our mind? Small intestine. And which part of the small intestine? That is actually ileum. Remember, that has actually villi structure. So here we may name it ileum, or we may also call it a small intestine. Okay, and then we have to name the structure. So a structure that is actually representing a small intestine is this L. Okay, understand? So we refer here. What is the letter? Letter is actually L. After that, next one is storage of bile. Okay, remember that bile is produced by liver and stored in gallbladder. So where does storage happen? Storage happen in gallbladder. So I'm going to write here gallbladder. Make sure the spellings are correct. And what is the letter? That is representing gallbladder in the diagram. Let's see gallbladder, where it is. Okay, gallbladder is here. This is actually gallbladder, which is represented by O. Okay, so this is what, sorry, this is what I am going to mention here. Okay, so what is that? Gallbladder is actually O. After that, what do I have? Next is chemical digestion of protein in an acidic pH. Whenever we think of acidic, something acid, we know that where it is produced in the stomach, HCl. Okay, and which is the letter that is representing? That is actually J. You can refer back to the diagram. So this is how we need to fill up the table. Now let's move on to the next part of the question. Okay, so next part of the question is this, which is actually asking us, okay, when we look at this diagram, what are they asking? Fat is particularly difficult to digest as it is not water soluble and forms spherical globules in the alimentary canal. So figure is a diagram showing what happens to fat globules when mixed with bile. Okay, so whenever we are talking about fat globules and see like how large molecules are converted into smaller ones, this is actually called emulsification. This is the key word that you are going to use in the exam. So what is that? This is actually emulsification. And why emulsification is done? Emulsification is done so enzymes can work on it. Okay, emulsification is a mechanical process where actually large molecules are converted into smaller ones. So enzymes, what is the enzyme that work for fats? That is actually lipase. Okay, so it increases surface area for lipase to work on these molecules. So here I write emulsification for 1 mark. Okay, so this is my answer here, emulsification. And after that I move on to the next part. Okay, what is the next part? Yes, okay, so the next part that we were just discussing, they're asking about the same thing. Explain the advantage of the process shown in figure. So what is the advantage of it? What will it do? It will increase surface area. You have to answer for 2 marks. So how you are going to get the answer for 2 marks? You are going to get the answer for 2 marks is, first mark is for what? It increases surface area. So this is what? This is my first mark. And where is my second mark? For what? For the action of lipase. This is my second mark. So here how I will go for 2 marks for this question. Understand? Okay, now let's move on to our next part. Okay, so what are they asking? They are asking about complete table to show how the uptake of glucose by cells and the concentration of glucose in the blood respond when the 2 hormones are secreted. So here what are they asking in the table? Okay, insulin. When they are talking about uptake of glucose by cells, so what will happen when they are taking more glucose? So insulin, what will happen? It will increase. And concentration of glucose in the blood, what will happen? It will decrease, since insulin is doing what? It is converting this glucose into glucagon. Okay, so this will be converted. So here let me write the answer for you, that what will happen. So here I am writing the answer for CRE. Okay, what is the answer? The answer is that uptake of glucose by cells. So what will happen? Insulin increases. Okay, and then next, the concentration of glucose in the blood, what will happen to it? It will it decreases. Okay, then next, what will happen to glucagon? Okay, so when I'm talking about glucagon, uptake of glucose by the cell, so what is happening? Okay, since this glucose is converted into glycogen. Okay, so this glucagon, what is happening? It decreases. Okay, and concentration of glucose in the blood. Okay, you can give actually two answers. Either it is going to stay the same, or you may also talk about that it increases. Okay, after that, when we are moving to the next part, state another hormone that influences the concentration of glucose in the blood. Okay, other than this, other than insulin and glucagon, what is the other hormone that works for it? And that is actually adrenaline. Okay, so this is the answer for C2. And what is that? That is adrenaline. Understand? Okay, so after that, explain why the control of the concentration of glucose in the blood is an example of negative feedback. So this is the question which actually we have got from the topic homeostasis. So what are they asking? That explain why the control of the concentration of glucose in the blood is an example of negative feedback. Okay, and negative feedback, first of all, what will happen? Any change in concentration, it will be detected. Okay, so first of all, because this is for 3 marks, so how you are going to mention it? You are going to mention about it that it is going to, let me write it down. Okay, so how you are going to gain 3 marks? First of all, change in concentration is detected. What does it mean? That whether it is going low or whether it is going high. Okay, so if it is going low, what will happen? Glucagon would work. If it is going high, insulin will work. Understand? Okay, so this is our first mark. And after that, of course, you are going to answer accordingly. For example, if you are going to talk about that change in concentration is detected, which change is detected? Increase in glucose. So what will happen when increase in glucose? So what will happen? You are going to talk about insulin. Okay, that insulin converts glucose into glycogen. Remember that glucose is a small soluble molecule, while glycogen is a large insoluble molecule. Okay, so after that, what will happen? It brings the concentration of glucose to normal. Okay, and this is the example of what? So the first mark, then here we have the second mark. Okay, after that, here converts glucose into glycogen. This is our 1 mark. And in fact, here we can, this one here I mentioned, it brings it to normal. And this is the example of what you may also talk about, homeostasis. Okay, that is also going to even give you 1 mark. So you have about 4 marks, out of which if you mention any 3 valid point, but I always recommend you to talk about at least 1 more sentence, so you are on the safe side to score more marks. Okay, so this is how we are going to answer such questions. I hope you like the video. I'm going to do more topicals on different topics. See you. Thank you.